Now, let's continue on the two proteins that the flu viruses are probably most known for, hemagglutinin and neuraminidase. Okay, so hemagglutinin is the type of protein on the envelope surface that binds the particles to susceptible cells. So it is also the major viral structure or antigen that antibodies are directed against. So the third function is that it fuses the membranes of the virus and the host cell. So the H protein is divided into two subunits, HA1 and HA2. The HA2 subunit is anchored to the membrane with a tail that extends to the cytoplasm inside the virion. Outside the surface, it extends as a stalk where the HA1 subunit is attached to it. So HA1 is globular in form and contains 5 antigenic sites or regions. So when we say antigenic, it means they are capable of causing or triggering an immune response, okay? So in the form of antibody production. So these regions are the ones that undergo mutational changes, no? Causing differences between strains and making vaccination difficult. The HA2 subunit also has a fusion peptide that is buried beneath the membrane. So this will be used to fuse the viral membrane and the host endosome membrane later when the virus has entered the host cell. So initially, 1 unit of hemagglutinin is one single unit called HA0. It only splits into separate subunits once they are split by proteases. So we will talk about this more of this in a moment. Okay, 3 units of the H protein form a trimer acting as one spike. So the 3 units make the spike more stable than just 1 unit per spike. So upon entering a host, receptors on the H protein attach to the sugars present on the surface of the host cell. The sugars in particular are sialic acid sugars. Remember that initially hemagglutinin is still one single unit. With the help of specific proteases present around, this single unit is split into two separate subunits, no? HA1 and HA2. So these subunits we already mentioned earlier. So they are split, but they are still connected with a disulfide bond. Okay. So this is important to activate the fusion peptide, which can be found at the end terminus of HA2, which initially, when it was not yet split, it was not activated yet, right? Because it's still stuck between the unsplit H1 and H2, or HA1 and HA2. So again, the splitting or cleavage of HA0 into 1 and 2 is important for the virus to be infectious. Without the splitting or without the cleavage, the fusion peptide will not work. Actually, at this point, the fusion peptide is not yet fully operational pa, because although it is activated, it is still hidden. I mean, the HA1 globular unit, diba, is still in the way. Okay, so it still cannot work. But at least it's activated now. So through endocytosis, the viral particles are then taken into the cell and packed within endosomes. So let's just make the host cell bigger na lang. Now, the endosome has an acidic pH. The acidic ions then enter through the M2 ion channel protein into the viral particle, no? And then this triggers a conformational change. The conformational change is that the HA1 subunits now bend, allowing the fusion peptide, which was hidden at first, and now it is able to attach to the endosome membrane, causing it to fuse into one membrane, which will then open and and then release the viral RNA segments into the cytoplasm, which then enter the nucleus. Okay, so this makes the influenza virus unique from other RNA viruses pala, in that the replication occurs in the nucleus. The replication of the other RNA viruses occur in the cytoplasm. All right. By the way, as early as 3 hours post infection, the virus is already able to terminate or stop the nuclear operations in the host cell to prioritize viral RNA replication. Inside the nucleus, the RNA polymerases transcribe the negative sense RNAs into the positive sense. Remember the RNA polymerase complex made of three proteins, di ba? At the end of the RNA segments. So that's why it's called RNA-dependent RNA polymerase, because they are actually found at the terminal ends of the RNA segments they transcribe. So anyway, the new positive strands can now act as mRNA, which are transported into the endoplasmic reticulum in the cytoplasm for translation into new viral proteins. So what viral proteins? The specific proteins that they were assigned to code for, di ba? So new nucleoproteins, new PB1, PB2, etc. Okay? And ultimately, new components to assemble into a new virion. All right. The nucleocapsids are assembled in the nucleus and then they move out to the cell surface. After new progeny virions are assembled and mature, they bud off the cell. But remember, they also contain hemagglutinin, of course, di ba? And because of that, they actually attach and bind to the sialic acid on the surface of the host cell where they came from. So how will they transfer to other cells kung nakakabit pa sila sa original cell? So this is now where the other important glycoprotein comes into play, which is neuraminidase. So neuraminidase is the other glycoprotein found on the surface of the flu virus. It is also antigenic. It is actually a sialidase enzyme that removes sialic acid from glycoconjugates. In other words, it causes the new virions to be released from the host cell by cutting the sialic acid connection with the virus. So it also helps prevent aggregation of the virions by removing the sialic acid residues present on the viral glycoproteins. Once released, the new virions can now infect other susceptible cells and multiply even more. Now remember, when looking at the whole name of type A flu viruses, at the end we will find the subtypes, right? Which are determined according to the HA and NA proteins, right? So for example, H1N1 or H3N2, right? So there are actually 18 different types of hemagglutinin proteins and 11 different types of neuraminases. So since they are proteins, the differences would be according to the amino acid sequences present. Okay, so there can be a combination of any of the H1 to 18 or N1 to 11. Actually, according to the CDC, there are potentially 198 different influenza A subtype combinations, but only 131 subtypes have been detected in nature so far. No? And so far, although there are a lot of different influenza virus subtypes, the most common that routinely circulate are the type A H1N1 and the type A H3N2. Type A is responsible for most cases of influenza epidemics. No? The Spanish flu in 1918, which was H1N1, and then in 1957, the Asian bird flu, which was H2N2. And then another bird flu broke out in 1968 in America. This time it was H3N2. And in 2009, another flu outbreak occurred in the US. It was an H1N1 virus believed to have originated from swine, but it was totally new and it was different from other previous H1N1 viruses. So, aside from affecting humans, type A is also known to affect birds. So, we have bird flu and other mammals like pigs, no? So, swine flu and even horses and bats. Okay. Type B is known to only affect humans. That's why there's no need to indicate the animal of origin, unlike type A. Influenza B viruses are not subdivided or not divided into subtypes, but instead they are further classified into two lineages: B. Yamagata and B. Victoria. Influenza B viruses generally change more slowly in terms of their genetic and antigenic properties as compared to influenza A viruses. Both type A and type B can be further subdivided to clades or groups and subclades or subgroups based on the similarity of their HA gene sequences. Type C influenza viruses have been isolated mainly from humans and in China from swine, while types A and B have HA and NA glycoproteins on their surfaces. Type C has only one multifunctional glycoprotein, no? The hemagglutinin esterase fusion protein, or the HEF. And if you remember, they only have 7 RNA segments, right? So they do not have the gene that codes for neuraminidase. So that's why there is no neuraminidase in their glycoprotein. But they are still known to cause upper respiratory tract disease, only milder.